Well, good evening and welcome to this ICT for Life Sciences Forum presentation. I'm Terry Cayley from NICTA. A big welcome to Brian Litt. This is Brian's first visit to Australia, one of many we hope, particularly to Melbourne. Great weather, you know, but great science too. And Tony will say a few words. It's good to have people who work in the area to introduce our speaker, so thanks Tony for doing that.
Good, thanks Terry. Yeah, welcome to the ICT for Life Sciences Forum. And it gives me particular pleasure to welcome Brian here this evening, mainly because there's very few people who could, I think, encapsulate what this ICT for Life Sciences is trying to do better than Brian does. Brian actually holds professorship in 2 departments, so he's actually split between neurology and bioengineering. And I can't imagine how he actually manages to balance both of those, answering to 2 deans and actually teaching in both of those faculties and with substantive appointments there. But it really does encapsulate what we're trying to do here to communicate and transfer that expertise between these 2 areas, the life sciences and the physical sciences. So Brian's area of expertise is epilepsy, and he works at a whole string of different things, many of which we'll hear about this evening. Brian originally did his degree, studied at Harvard, and then did much of his training at Johns Hopkins University. And he has a list of awards here that would be way too long for me to read out, but it's very, very impressive. I should just say that he's also, and you'll see here from his list of disclosures, he's been involved in actually helping found a number of companies, including Intellimedics and BioQuantics, as well as consulting with a number of companies, including Neurovista, MC10, Medtronic, KCI, and also licensed technology to all of these companies and to NeuroPace. So you can see that he's actually had a very, very big impact upon this field. It gives me particular pleasure because Brian's one of those people whose papers I'd been reading for years and years, and so to have him here in Melbourne is a great pleasure. Brian, we look forward to hearing your talk.
Well, thanks so much for the wonderful introduction, Tony. It is my first time in Australia, and I've been really amazed with what I've seen, not just with the warmth of all the people, but really the vitality of the university and the communities here, both in medicine and in engineering. We'll cover a bit of ground today, and I'll give you at least one perspective on how we're trying to look at epileptic networks and develop new therapies for them. As Tony pointed out, that I work with a number of companies. I don't go out and give talks for them, but we work on licensing technologies and collaborating. That means that, you know, I could potentially have some conflicts, and I'll try to point them out, and I'd like to think I'm my own harshest critic for the work we do, but it's important to be aware of these things. It's also important to acknowledge that when you do this kind of work, I may be the person who's speaking here, but it's really the work of a lot of different people, trainees at all sorts of levels, and since they all can't be here to enjoy your hospitality, I should just point out a couple. The lab I run really sits in a dry space. It's about 3,000 square feet, where we bring together people from a variety of disciplines to work together. We have MDs and MD PhDs up top here. We have graduate students and postdocs in engineering, and then a number of undergrads, master's students, and then collaborators in a variety of areas. Material science, John Rogers at Illinois, Sherry Kagan at Penn, a number of physicians in neurosurgery, neuroscientists like Diego Contreras and Greg Worrell at the Mayo Clinic, child neurologists, and I'll try to point out other folks as time goes on. Well, the world we live in epilepsy is one in which there's kind of the practical space we live in, and it motivates us a lot for trying to make changes in the way we do things. So what we do is if you have mesial temporal lobe epilepsy that comes from the hippocampus on one side, the very best therapy available is to take a brain that looks like this and turn it into a brain that looks like that. It looks pretty crude, and frankly, I think it is. But it's very effective in the right cases. The analogy I like to give is that You have a problem with the lights on in your house. They're blinking on and off, and you can't really localize where the problem's coming from because all the wires are sealed up in the wall. So you figure out finally that it's coming from the garage, so what you do is bulldoze the garage. It's effective, but it's not the most elegant solution, and you lose a few things at the same time. And so what motivated me when I started in medicine, I didn't do my scientific training before, but rather went through as a clinician and did all of that, and then realized that the therapies that we have a lot of faults to them, and then went back and into full-time academics from really a more clinical, full-time clinical appointment. The sensors we use and to localize epileptic networks and the approaches we use are actually pretty old. These are standard electrodes that are used to be implanted in the brain, grids and depth electrodes that use centimeter-spaced electrodes. The techniques were established at the Montreal Neurologic Institute, and they're well described by a book by Penfield and Jasper. It's called Epilepsy and, I don't remember the full title, Epilepsy and the Functional Anatomy of the Human Brain, or something close to that, published in 1954. And so it's important when I talk to students to say that they should really question why we do things. And when I asked why we sample the brain at centimeter spacing, why we put in electrodes through burr holes underneath the temporal lobes on top, depth electrodes through the occipital approach along the hippocampus, or perpendicular in stereo EEG like they do in Europe, or putting in electrodes on this scale 1 centimeter apart, the reason is because of tradition. When you think about the neuronal networks that are involved in neurologic disease, they really are on a much different scale, a number of orders of magnitude smaller. And the reason we use this technology, it turns out, is because that was the limits of the technology when the techniques were developed. And people haven't really proven so far that recording at other scales is more useful. We think it may be. When we do functional mapping of the brain to figure out what you can resect and localize where seizures come from, we build maps like this routinely. We do 3D reconstruction of the electrodes that are placed, and we stimulate between pairs of electrodes. There are a couple of articles that establish what safe parameters are for current densities to inactivate brain or to activate it, depending upon how you stimulate. And the theory is that when you stimulate at low currents, on the order of 1 to less than 5 milliamps, that you're activating the neurons in the deep layers, 3 and 5, that are beneath these electrodes. The function you get at low thresholds tell you that that's the function in that area, and if you damage that region, you're going to lose the function. So these are the kind of maps we make, but it turns out that the dynamic plastic networks of brain that give rise to these functions and to pathologic networks probably aren't so simply described. When we do surgeries for epilepsy, the idea is that the clinicians, usually the neurologists, electrophysiologists, record from electrodes in the brain, and they pick up things like these interictal spikes, that's between seizures, primarily on ones that spread to the other side in this patient. This patient's seizures began with a big single discharge in the blue electrodes, then a fast gamma buzz, so maybe something on the order of 30 to 40 hertz in the yellow circles, then lowered the frequency as it chirped down to on the order of 20 hertz and what we call a beta buzz in the red, and then the seizure was sustained here. We give these maps to the surgeons who go into the operating room, and then they try to adapt what we've shown them to doing an operation. But they run into all sorts of technical issues. There's a blood vessel here that they're concerned about. They're concerned about removing an area which may have a different kind of functionality, or resecting white matter that goes beneath a particular area, so they modify the surgery. And indeed what they remove, as denoted here in green, and this is an extreme example, might not be the region exactly that we want them to remove when we do the electrophysiology. The fact is there aren't good standardized methods between centers. If you went to 5 different epilepsy surgery centers, you would get subtly different 5 different surgeries. So, I mean, this is sort of the state of the art where we are right now. There have been a number of approaches. Epilepsy surgery has been around since the 50s. The idea is kind of localize, record where the seizure seems to begin, and then figure out the function of the brain around that region, take out the seizure-causing areas, leave the good brain, and the patients will be seizure-free. But the fact is, unless you have a focal lesion that's clearly the cause of the seizures, like hippocampal atrophy or a focal cortical dysplasia or an abnormal blood vessel, that the chances of being seizure-free can be quite low, like 40 or 50%. Now, the patient's improvement is actually usually substantially more than that. But even 40 or 50% for people, chance of being seizure-free for people who on another medicine have less than an 8% chance of being seizure-free is worth a shot for many of these patients. But it's still at at a level where there's no guarantee of success and there's substantial morbidity. In recent years, there's been an interest, particularly from people who are successful in the cardiac device industry, to apply these same principles to brain. There's a long checkered history of stimulation devices. A lot of it arose out of controversial experiments that were done in parts of the world. Mexico City was a place where a lot of pioneering work was done, but the results and their statistics have often been questioned. But as a result of that work, Medtronic, for example, did a trial of a device where electrodes were placed in the anterior thalamic nucleus because of studies that showed you could reduce seizures in animals given convulsant agents if you stimulated in these areas. So they implanted on the order of 200 patients over a period of a few years and showed that there was benefit, but it wasn't a home run. The FDA did not approve that device in the United States, and they've called for a second trial. They think because the first trial didn't go out long enough. Similarly, people from the defibrillator industry developed this device that was promoted by NeuroPace, and we licensed 3 patents to NeuroPace. They detect seizures on EEG. The patents we licensed to them were really just features that can be used to do very rapid seizure detection. 3 orthogonal features. One was a power feature, one was a frequency-based feature, another one was a signal complexity feature that could be reduced to analog circuits that didn't require much power. The idea is you detect the seizure onset and you stimulate to turn it off. Sounds like a good idea. The clinical trial with that device was fair. Now, the company didn't require in the trial that the seizures be well localized. They just said that if they were in the general region, putting the electrodes there was probably good enough. I personally think that that's probably not the best approach. But often when companies spend $100 or $120 million on a clinical trial, they want to get the broadest approval from the regulatory agencies for application, and they felt that limiting it to just where you knew pretty much where the seizures were localized to would cut their market way down. It turned out to be not a wise decision. The FDA, about 3 weeks ago, decided not to approve that device and again called for a second clinical trial. Does this mean that devices for epilepsy and implantable neuro devices are not worthwhile? It doesn't mean that. It means that there are market pressures sometimes for people to run to do a clinical trial of a device before the basic research has been done to really establish the best way to use it. And there's a big balance. We may never know mechanisms of some cardiac arrhythmias because the defibrillators work well enough that there's not pressure now to go and do more basic science there. Anyhow, so these are technologies that are a work in progress. Realize that this device is like the first pacemaker. The problem is now it's harder and more expensive to do trials. Speaking of questioning the status quo, when I was a fellow learning epilepsy, I was taught this is an intracranial EEG recording from a grid like what I showed you put on the brain, and I was told this is the signal before the seizure, each one of these channels from a different electrode, that there was a desynchronization of brain activity here, and then the seizure started with this rhythmic synchronous activity here, and everybody knows that seizures are rhythmic synchronous events, supposedly, at that time. We did a very simple study with Greg Worrell, who came to the lab from Mayo and then went back. which showed that it turns out that this is really high frequency activity that's outside of the bandpass of the device. That when we record clinical EEG digitally in epilepsy monitoring units, it's from 0.1 to 70 hertz typically. Now, some device manufacturers said you can sample at 500 hertz, 600 hertz, like the device that we have in our unit. Except what they didn't tell us, which we had to figure out by looking at the signals that we input and then recorded, is that they had a hardwired anti-aliasing filter at 100 hertz on the device. So you could sample till the cows come home, but it didn't matter. So these kinds of assumptions. Oh, and I asked a bunch of the old guys, why is it? Ernst Niedermeyer is the one who taught me EEG. He passed away a few months ago. It was a big loss, kind of one of the gods of EEG if you're in that field. And I said, Ernst, why is it that they made on the EEG machines, which they transferred when they went from paper right to digital, 0.1 to 70 hertz, right? Why is it 0.1 to 70 hertz? And he looked at me with this Austrian accent and said, well, it's because above 70 hertz, the ink splatters all over. So if you're doing engineering in the hospital, the fact is that there are a lot of things like that. All right, so what are we going to do? Well, if you're Sid Cash at Harvard, you're gonna, or Catherine Siobhan at Columbia, or the guys at Utah, you're going to take a Utah array, now called the BlackRock array, and you're going to pop that fella in. It's 4 millimeters on the side. It's a bed of nails. It's 100 electrodes. You sit it on top of the brain. You take a little air hammer and go pop, and it pops into the brain. It's not pretty. Fortunately, they're asleep. And you're going to record neurons. So that's a great idea. Neurons give you the language of cortex. They're going to give you the language of seizures. And you record 2 seizures. And here are the rasters. Each one of these numbers are the cell number. So there are 130 cells here, something like that. And this is over time with a seizure. And what do you see? You see that some of the neurons, their firing rate, each little tick here is a spike. Some neurons in the seizure, the firing rate goes up. Some of them it goes down, like these guys, and some of it just stays the same the whole time. These are 2 seizures back to back, so it's kind of reproducible. But does that help you? Well, it's difficult, right? So this scale, the neuronal level, might not be good unless you could record from all the neurons in the brain at one time, or at least over a large clinical region. When we put those grids in, those are huge. They're 8 centimeters by 8 centimeters, and sometimes we put a couple of them in. So I don't know about you, but if somebody was consenting me for a surgical procedure and said, we are going to put 5,000 nails into your brain, and we're going to pop them with an air hammer, like that, you'd probably say, ooh, that's not a good idea. I mean, people get epilepsy from lesions like that, right? And so there's a big controversy about that. The analogy that I like to use is that in figuring out the scale, if you're looking at a group of people on the street in Manhattan, and they're milling around, and there's a lot of stuff going on there, looking at EEG, signal averaged over large volumes, is like hanging a microphone from a helicopter and trying to figure out what's going on in the street. Is it a riot? Is it a celebration? Somebody's birthday? Are they setting the town on fire? What's going on? So instead of that, lower 100 different microphones down and talk to individual people. Well, how are you? Well, I just came out to get a drink, you know. No, I'm turning over a car. I'm robbing a liquor store, you know. It's not going to help you unless you can sample broadly. So we don't think EEG might be the right level. It's not clear that units are the right level because of sampling error, and we focused on local field potentials, and I'll talk to you a little bit more about that. So this guy, John Viventi, came into my lab. He was very precocious. He'd finished a master's and a bachelor's degree in electrical engineering at Princeton in 4 years, while like running the cycling club and singing in the glee club and all this other stuff. Founds a startup company that's eaten by Qualcomm a few years later, where he got a real education about business because he helped found this company and got 0 for it when it was acquired. And then worked there for a while, and then came in and said, I want to do a PhD in the lab. He said, what do you need done? I said, it's really simple. We have brain that we need to record, you know, down to the micron level. And we have electrodes that look like this that were invented in 1950, and we don't think they're good enough. I want you to take one of these and build me a device that has 10,000 electrodes on it. And that'd be great. And he said, that's impossible. And I said, you'll figure it out. And left him. And this is what he built, and I'm going to show you about that and why we think we need it. So here are the questions we focus on in the lab. I've set myself a timer here that'll give me 15 minutes, so I'll have a few minutes to finish up. When you hear the timer, it's just me, because these guys are too polite to throw rocks. Actually, Mark Cook is not too polite to throw rocks. So what's a seizure? Okay, you could say we brought this guy from Philadelphia, paid for a plane ticket, flew him halfway around the world. Actually, it's more than half, isn't it? It's a 14-hour. Anyhow, we flew him a long way. He's supposed to be an expert on epilepsy, and he says he doesn't know what a seizure is. I want my money back. They didn't charge you for this, did they? Good, excellent. So it turns out that if you see a recording with one of these rhythmic events that lasts a couple of minutes on EEG, and the patient's like chewing and having picking movements or automatisms or dystonia or convulsion, then you could say that's a seizure. I know what that is. But suppose they just are bland when they have the seizure. We have an epilepsy monitoring unit. We do continuous video EEG. When the patients are not having seizures, they're looking at the TV like this. When the patients are having seizures, they're looking at the TV like this. And so it can be pretty hard if the behavior doesn't tell you what's going on. But if the discharge isn't 2 minutes, suppose it's 1, 30 seconds, 3 seconds, 100 milliseconds, when's it a seizure? It's not such a simple question. We're trying to figure that out. What's the basic unit of brain that causes epilepsy? There are a lot of people much, much smarter than I am, and I'm not a great basic scientist, who have looked at this issue. Barry Connors at Brown says that it may be just a handful of layer 5 neurons together that are firing abnormally that constitutes a seizure, but there's still a debate about this. And then we want to know the topology, architecture, scale of the epileptic networks, and then how to intervene. There are 2 ways to go about this, and you have fantastic examples at the epilepsy centers here and neuroscience and engineering in Melbourne. We have people working with patients, and they're doing clinical work, and they're trying to just improve the clinical devices to get the answer to these questions. And then you have people who are working with rats and mice and cats and dogs and sheep. I've seen all of that with epilepsy, trying to do basic experiments on the cellular level to figure out and reach up to the clinicians to find the answer. And somewhere in the middle is probably the right way. So that's the kind of stuff we do, and this is what we focus on in the lab. We build hardware. We do a lot of work with algorithms. If we're building devices that have 10,000 electrodes that are recording at a kilohertz or a couple of kilohertz from each channel continuously for 2 weeks in the hospital, that's a lot of information. So we spend a lot of time with the statistics and the machine learning community to try to boil that information down. We do human work. We collaborate with animal electrophysiologists, and we do some computational modeling. Biophysically accurate models, mostly in neuron. We work on devices, and then we have this idea, and one of the things I'm trying to sell to your community here is to work with us on an international data sharing facility that we have funded through the NIH. It's free. I know people come from America, they're like, buy this. You don't have to buy it. It's a way to, and a philosophy we're trying to work on for science, where we have common data sets, people analyze it, they post their annotations, their results, their code for analysis, and then work together to solve significant problems, like what's the best detector for high-frequency oscillations? Publish that together, and then you have the data there, you have the algorithms, and if you want to validate the research, you can log on and run it. There's a lot of problem with validation of research in our field. So more about that maybe later. And this is just the idea that, and I've heard people say this at the Melbourne Epilepsy meeting this week, that the incentives for research are mixed. If the measure is your H-index, how many first and last authored papers you have, how many grants you have, there is no incentive to share what you're doing with anyone and advance the field. It would be much better if people were credited for their impact on advancing a field. And I've said this, I served on a panel for the NIH a year ago where I said that, fine, have people compete for grants and give scores and give half the grants that way, or maybe 2/3, but save 1/3 of the money for people who are doing spectacular work and give them another 4 years as a prize. so that they can spend most of their time doing research rather than just writing grants. It's just a thought. It's the reason why I don't have a high administrative position. I wanted you to know that. Okay, so Greg Worrell came to the lab, and we were interested in trying to get higher resolution recordings. And he got this idea to put 40 micron micro wires in these little tufts between these centimeter scale electrodes, and convinced AdTech, one of the manufacturers, to do it. They're dear. They charge a lot for it. They're put in by hand by little people with little fingers. They actually make these on a jig, seriously. And also the people at UCLA, you know, Chuck Wilson, Yitzhak Fried, Anatol Bragin and his group had put out these Banky-Fried electrodes with microwires, so it sounded like a good idea. And what we found is a couple of things. We started to notice, as Anatol Bragin had published at first in KNA-treated rats, that you see high-frequency oscillations in these animals around the epileptogenic region. And we found in these 7 patients, these are the number of high-frequency oscillations, and I'll define them better in a minute. And they came detected in black by the microwires. There were more than were detected on these big macro electrodes. So that's interesting. And also the epileptic temporal lobe in each one of these patients generated more of the high-frequency oscillations than the non-epileptic side, which is the shorter column. This patient probably didn't have temporal lobe epilepsy. The problem is, how are we going to mark these high-frequency oscillations? Because in Anatol's study, they did it by hand. They used a crude detector with very high sensitivity, and then they marked them by hand and filtered through a lot of events. So we got together 4 experts. I was one of the experts, 3 others in our institution, and we marked a bunch of high-frequency oscillations by hand. And what we found was that the other experts didn't know anything because we didn't agree. And so we started working with the machine learning people in computer science and started to do unsupervised learning. Now there's a literature that talks about ripples being like 80 to 200 hertz as being important to cognition, cognitive oscillations in memory networks. And then Anatol said that above 250 to 500 hertz, these oscillations were specific to epileptic networks. So Justin Blanco developed an unsupervised method for his PhD thesis, no big deal. We had him analyze 13 terabytes of data. It was huge, from 11 patients, 9 of them with epilepsy, and 2 of them who were control patients that had these grids put in as part of a stimulation trial for chronic facial pain. What do we find? Here's an example of an anecdote. A patient who had a tumor removed, who still had seizures. These are recordings from these electrodes and some of the microwires nearby. And what we see is that there are outlying regions, statistically, that have more high-frequency oscillations. It turns out that all of these turned out to be the gyrus next door, which was resected and the patient was seizure-free. Anecdote, N of 1. Interesting, doesn't prove anything, but it's interesting. So we looked at these high-frequency oscillations and found that actually they were seen in the regions around where the surgeons said the seizures began. And it became kind of interesting, and there's been a large amount of activity in the medical literature right now where people are using all sorts of different methods to map these high-frequency oscillations, and now saying that resecting them correlates with better outcome from epilepsy surgery, and they should be used as a marker for where seizures come from for epileptic networks. It turns out that there's not uniform agreement about the accuracy of some of these methods. Some of the groups that have published the most use marking them by hand as a measure, and we have some issues with that. We also think that with some of the filtering that's going on, they may be turning high gamma, just fast activity, and making it look like oscillations. So these are things we're working through. We also noticed that there were these events that looked like seizures that occurred on single microwires and not seen, sometimes seen, but often not seen a millimeter away on other electrodes in these tufts. So these are the macro electrode recordings from the centimeter electrodes, and these are the microwires. And I call your attention to these events that when you blow them up look like seizures. The clinical criteria we use is that they frequency modulate, they chirp, they either go boo, or they go boo, or sometimes up and down, and that they spread, and that they sometimes lead into seizures. And that's what we see. Here's a custom electrode macro electrodes in blue, microwires in red, and you can see 30 seconds, sometimes 60 seconds or longer. The fast activity begins on these microwires and then leads into a seizure, which then shows up on the blue electrodes. Here's kind of the bottom line in what we're seeing in patients who have these electrodes in. So this is channel number. This is time, about 40 minutes before seizure. This is when the seizure occurs, this double line here. These are the macro wires and these are the micro wires. And every one of these little ticks is an automatically detected micro seizure event. The pink is the region that the clinician said the seizures came from, and that's what was removed. We see that as seizures approach, and now we've analyzed the whole data sets, not just the areas leading up to seizures, but there is an increase in these HFOs often before seizures begin, and then sometimes a quiet period right before. Are you surprised if high-frequency oscillations define epileptic networks that when this area was removed, the patient was seizure-free for a few weeks and then started to have them again? It was actually a few months, 3 months. Well, if you believe in neuroplasticity and that epileptogenesis, the creation of epileptic networks over time, is an ongoing process, it's not surprising to think that these areas could generate seizures by themselves. What does that mean? It means that the technique from 1950 of removing a huge hunk of brain where seizures appear to start might not be the right approach. I'm not a rocket scientist, but these are things that really need to be proven in clinical trials. I can sort of make fun and throw stones at tradition in medicine, but the fact is we do care about our patients, and if you're going to suggest a better technique, you have to prove that it works in a trial. So maybe it would be better to go and run some kind of detector or device over this and ablate every one of these regions that's generating a micro event. Maybe better to have a device that sits on here and sees them and tries to turn them off electrically. It's not clear, but this surgical approach probably isn't the best thing, that epilepsy comes from a network, not from a focus or a spot. The other thing is that these imply is that regions that generate seizures may be on the order of a few cortical columns, not 3 to 10 cc's of tissue like I was taught when I was a fellow. It makes you wonder about these things. So we go back to John Viventi. There he is. He was smiling when he started. By the time he finished in my lab, he was pretty beaten up. That's not true. And then some of our major collaborators here from University of Illinois. By the way, it's really helpful to do research with a guy who's won a MacArthur Award in the US, because some of the journals we've submitted the papers to that I'll show you wouldn't have given me the time of day if John's name wasn't on these papers. I've never gotten a call from Science that said, well, we'd like to put you in our new journal, Science Translational Medicine. Would that be okay if we give you cover space? So those are calls to John Rogers. I don't get calls like that. So John invented this technology where he builds circuit elements like you would on an IC and connects them with these flexible silicon nanoribbons. They're kind of scrunched up on here so that they can be pulled and stretched in a variety of directions, and the interconnects, they stretch and go apart. We build them in layers like you build regular integrated circuits, and then strip them off the top of the silicon chip and put them on these flexible sheets of rubber, plastic, things like that. So here's the first one. That's Viventi. You can see him labeled there. And I like to show this first one because it shows the first error that we had, that sealing these things has been quite difficult. This is sealed in SU8. These have, each one of these is a contact. They have circuitry on them right next to the contact, an amplifier for each one of these elements, and then multiplexing circuitry. These are multiplexed just row and then sampled, but we can double multiplex them so we can reduce the wires quite a bit. And the idea is to have a USB or something like that come out. This just shows that when the insulation fails, you get hydrolysis of water, and we did have flame come out of the back of the animal's head in the first experiment. It was actually somebody else's experiment that we piggybacked on the end of. Of course, I was right there with them in bed when they called because we had to wait till the end of Diego's experiments, which ended at midnight. So my team would drag the instruments across campus in this thing that looked like a coffin in the middle of the night to go and do these experiments. John's very dedicated, actually. He's now on the faculty at NYU, and he's wonderful. And this is what a failure looks like. It's a little geyser of electrolysis coming off of that thing, and we hate to see this stuff. This is what the devices look like now. And we can roll them up and put them in catheters like this. And we're doing experiments now where we're rather doing these big craniotomies like I showed you. We're making burr holes and trying to put them in endoscopically. And I'd mentioned at a previous talk that I have a very aggressive neurosurgery resident who's doing his research here in my lab, and he wants to put these on the body of the hippocampus through the ventricle by going through the back of the eye. And it turns out that they do procedures like this. We're doing them in dogs first, and it turns out I asked him how he was going to perfect this technique, and he said basically that he had ordered a bunch of cadaver heads that he was going to practice on, and that he had kind of forgotten to tell me that they were going to roll in any day now. I hate when they do stuff like that. All right. So we first tested these things on the heart because there was noise in the original devices, and the EKG has a much larger signal, a few orders of magnitude higher, millivolts rather than microvolts. And it's very nice. They flex as the heart beats. And rather than, if you have a cardiac arrhythmia and they need to map you, they take one of these things, they thread it around either through the chest wall or up inside the heart, and they have these wonderful 3D localization systems that know where the catheter is in 3 dimensions. They record here for a little while, freeze the location, then they move it, freeze it, and they put together this little puzzle piece of where they are. But we can record waveforms as they move across the grid, and we have great movies of it starting here in red and ending up in blue. So that's what an array looks like on the brain, and I'll need to speed up a little bit. 720 channels going down to about 30 wires. These are the platinum-coated ones. We also put them on biodegradable silk, so that, which can be programmed in its material content to melt and allow it to go down and settle into the brain to give us better contact. So the silk actually, on top of the silk, we have 2 to 5 micron lattices. It's like a cross-section, like the streets here in Melbourne, in a grid. And the silk dissolves and it lays down onto the surface. And this is kind of what it looks like when it lays down there. And you can see with this visual evoked potential, as the device melts, signal quality measured in green and yellow is good. The gray are reference electrodes, and ground really improves as they get closer to the tissue. So let's show you what a couple of these recordings look like. This is a cat brain. I apologize to you if it's offensive, but Diego works on these animals, and we really try to get the very most out of them we can. He does his experiments, and we come in later. And so in this occasion, we're recording across a sulcus between V1 and V2 in the visual field. And this is what the visual evoked potential looks like when measured from one of these arrays. Rather than recording a single waveform, it's really like a voltage-sensitive die. Each one of these has a stimulus in a different part of the visual field which corresponds to this whole screen. And so in this experiment, what we published was just demonstrating the somatotopic organization of the visual cortex corresponding to the retinotopic organization, corresponding there. This is another experiment where we put the array in a slightly different region near the occipital area and then put some picrotoxin focally next to the grid. And then we induce seizures. It's a GABA blocker. And this is what 10 channels look like of the seizure, but it's really hard if you read this to understand the spatial characteristics. We think of EEG as a bunch of lines, like reading music from a piano that you read in parallel. But in 2 dimensions, this is what a seizure looks like. This is the average of all of the electrodes here. This is about 400 of them here. And what you can see is waveforms come across. Red is depolarization, blue is hyperpolarization that follows afterwards. And you can see these rhythmic patterns. So there are a few beats here, and then it's going to change direction in a minute. And then it establishes this prolonged pattern for the seizure. And so we looked at these things, and we started to analyze them. And you see this repetitive pattern that goes around in a circle. And to us, they look like spiral waves, and that's what we're modeling them as. And it turns out that folks here in Melbourne, Dean, who's sitting in the audience, has also found these in some recordings of human EEG recorded in a different way with the Utah array. So they've been described in hippocampal slices as well. But they haven't been, I mean, if actually I need to see these human recordings because it would be really important to us if these patterns were really what we see in human epilepsy. Because this is contrived, right? This is a acute chemoconvulsant that's given to the animal. And as this pattern goes on, and it's kind of mundane, I'm just going to take you to the end of it quickly and let you watch it. It's going around counterclockwise, and then watch how it ends. There's another wave that looks a little different that turns out to be in the opposite direction, and that stops the seizure. So interesting. And then there's another wave that's looking to start it all over again. So what we did is took a look at the individual spikes, detected them, and made delay maps of them moving across the grid. And this is the way we summarize the delay map. So it starts in red and moves out to blue. I'm sorry, the colors are reversed here. And then we looked at patterns of spikes leading up to seizures. And what we see is if you take the different spikes, PCA them down to a few components, cluster them, use a measure to tell you what number of clusters is optimal, at least by one technique, using this Tibshirani gap statistic, you see that the spikes aren't random across the grid, that they fall into clusters. And in this case, in this animal, we found 17 different patterns. I don't know that the exact number is important so much as to say that there's a repertoire, a finite number of patterns where these epileptiform discharges spread in waves across the grid. So this one goes blue up to red. This one goes blue around. And what we saw leading up to the seizures, and we're trying to validate this now in more animals, is that you see these waveforms kind of come across and then start to turn, and then they turn into the spiral pattern which stays. And then I'll show you actually what we plotted out a seizure as. So here's the one I showed you. It starts with a planar wave moving in this direction, which moves into a clockwise spiral. But then another planar wave comes in the opposite direction and changes the spiral into counterclockwise, which persists until another wave hits it, perhaps at the right phase, we don't know, because we haven't tried to measure it, and turns the seizure off. I have no idea if this is what happens in humans, but it's interesting in this model. And then another planar wave tries to start it and doesn't in this case. So Anne Van Leer, she used to be Chamberlain before she got married, is very interested in looking at these patterns and how seizures are initiated, because cardiac arrhythmias look just like this. The cardiologists call them rotors, but they're spiral waves that we see in ventricular arrhythmias, and they happen because they move around an area of anisotropy, an area that doesn't conduct right. So you see the wavefront hits it, one part gets slowed down here, the other one moves, and starts to go around because this tissue is not refractory yet and goes into this pattern around this anisotropy, and that's what causes a ventricular arrhythmia. I have no idea if that's what happens in human brain, but we know that in patients with cortical dysplasia, the seizures seem to come and arise around the outside of the cortical dysplasia. Typically, they're not in the middle of the dysplasia, depending upon the type. So this idea of a local anisotropy might not be so unreal. It's very interesting that an epileptologist would read each of these spikes as being the same discharge, but when you look at them in 2 dimensions, they're moving in vastly different directions. Hence the argument that higher resolution recording might be more important. This is just the idea of, so what is a seizure on the macro EEG, right? You see these waveforms, and it relates to like when, on the micro scale, does the seizure actually begin? You know, is it the waveform beyond which the spiral is going to happen? I don't know. But here's what we see clinically. We see a single discharge. That's a spike. That might not be a seizure. This has some after discharges, a few more, a second, 2 seconds, 4 seconds, 10 seconds. Which one of those would you call a seizure? Do you think they're all seizures? It's not clear. On the micro scale, we have the same problem. We're using some other techniques, and I'll gloss over these briefly, but we're very interested in looking at, on the micro scale, these small events, these small rotors or spirals, the longer ones, to try to understand their relationship to clinical seizure events. So Drawson Wolson is going to finish up this year in the lab, and he's very interested in developing new statistical models for clustering to try to look at these events and pick them up, because there are many thousands of them that occur in these prolonged recordings. And we want to know what's the distribution of these 1-second events, 10-second events, 100-millisecond events. How do they relate to seizures? And this is just an example where each one of these is a screenshot from a different seizure, and the borders on the outside are the seizures from a single patient. And you can see it's not perfect, but he clusters them pretty well in an unsupervised fashion using this method, and he's working on that more. We're using a number of animal models. We're using acute models like I've shown you in the cat, but it turns out dogs have seizures and epilepsy with the same incidence as humans, about 1%. They're refractory to medicines, meaning they don't respond at the same as humans, which is about 1/3. And their seizures on the intracranial EEG look just like the human seizures. So I actually presented these 2 dogs at Epilepsy Surgery Conference, pretending that they were humans, and completely fooled my colleagues. This is a patient with, and these are implanted with the Neurovista device, which really the first human implants in the world have been done here at this university, which humbles all of us in the States. This is a focal seizure coming from the occipital region in this dog. We have about 12 dogs that we have between Penn, Mayo, and the University of Minnesota with epilepsy who are recorded. And Kate Davis has been working on that. She's a clinician as well as an epileptologist, works in the lab. And here's one that's a regional onset seizure that's poorly localized. So we're hoping that doing some of these experiments in a natural epilepsy, like dogs, will really help reveal more of the physiology to us rather than creating chemoconvulsant agents. We're using some of these same devices. This is an experiment where we're collaborating with people who are interested in an auditory cortical prosthesis to be able to stimulate and record, and this is in a rodent, showing that we get very good agreement in this array between this optical image with its somatotopic or tonotopic organization of the cortex. Very good correlation here between the optical image and what we see recorded from the grid. There's an interest in potentially applying some of these technologies to visual prostheses as well, but we don't actively work in that area. Besides, why would I come to Melbourne and talk about that? We're very interested in adapting, so since these devices are made from silicon, we can put a variety of different sensors on them. Temperature, pressure, flow. We can put LEDs on for optogenetics. We can put them on other flexible devices. This is a project that MC10, a company that's licensed some of this intellectual property from us in Illinois, is working with Medtronic on new catheters for ablating atrial fibrillation. Where are we going with the technology? This work is clearly trying to go from the top end, right? It's trying to look at what we do clinically, find out what the best resolution is, and improve patient outcome by mapping epileptic networks better. But it's limited, right? We're only recording from the gyral surface. We could make penetrating elements, but it wouldn't be safe to penetrate enormous regions of brain like this. In the end, you know, as somebody mentioned in a previous lecture here, it's the three-dimensional problem that gives us pause, right? Because ideally, we'd like to be able to record from the sulci, where 2/3 of the cortex are, not just the gyral caps. We'd like to be able to get an image from many more regions of the brain than we can reach. We think that we may be able to improve outcome as it is with this incremental step, but in the end it's going to require something even more creative to get imaging functionally from regions of brain that we can't reach from the surface. And I'm not sure what the answer to that is yet. We're exploring with colleagues some nano constructs. I mean, it'd be great if you could build nano devices that could get across the blood-brain barrier and allow you to communicate with individual specific neuronal types. There's technology that's coming along that makes us think that that could actually happen. But it's going to require efforts from people like at your university, ours, many other good places to work together to make those sorts of things happen through incremental advances. For these arrays, for this technology. I think there's unprecedented potential to take this kind of work in the neurosciences, the kind of work you do here, the kind of work we do, and reach up to make a clinical impact. It requires exactly what you have here, exactly what this seminar is for, right? You've got engineers, medical people, neuroscientists, basic scientists, and many people in the same room, in the same buildings working together. We're working to do that better at our university, and I'm going to take back, when I leave on Saturday, stories of what I've seen here and said, you're not going to let them do that without us, are you? That's not going to go far, but we'll see. I think that it's important to keep a balance. Let the technical innovation drive you, but also let the clinical and scientific need be a driver. And these are the main challenges we have: scale, encapsulation, connectivity, immune response. Thanks.
Again, a big thanks, and thank you all for coming this evening, and again a reminder for next week.